Hey everybody, it's Mr. Eisens here. We are going to be looking at our cell membrane uh, and cell transport notes today. So let's get right into it. All right, so taking a look at our cell membrane, the cell membrane, remember, is going to be on the outside of the cell. Okay, here's an animal cell, the very outside of it, whereas the cell membrane inside a plant cell is going to be just inside the cell wall. Okay, so keep in mind that it's slightly different. Remember, our cell membrane is made up of proteins and fats. Okay, we call these lipids. And so what's going to happen is our cell membrane, remember, it's a meaning two biphospholipid layer. Okay, so it's got these two phosphorus heads on each side, and it's going to select what goes into the cell and what goes out of the cell. All right, so here we go. The plants have a cell wall, like we said. This is made up of cellulose and fiber. We really have a hard time digesting this, and we really can't digest it, and that's why it's fiber. Fiber doesn't get digested. It just goes through your body. All right, it is rather good for you. And then remember, bacteria and fungi also have cell walls. They do not contain cellulose. They're made up of something else. They're made up of peptoglycan usually. And then we also have, we'll go one back, cell membranes in the animal cell. So all these cell walls are porous, which allow things in and out. So just keep that in mind. The cell wall also allows things in and out, just like our cell membrane is going to do as well. So porous just means, think of like a sponge. It has a lot of pores in it. It's got these little holes that can fit things through it. So you can see all these different things are fitting through the cell wall, getting to the cell membrane. And the cell membrane is going to decide to let them in or not. Okay, and so that cell membrane is going to be basically what decides what is inside of the cell and what's outside of the cell. So the cell membrane is around the entire cell and only lets certain things in. Okay, it's selectively permeable, meaning it selects for things that it wants to come into the cell. Okay, and permeable just means allowing things in and out. Okay, so it selects what it allows in and what it keeps out. All right, homeostasis, we got to have an even flow of energy coming into the cell, or potential energy, so that the cell can do everything it needs to do to stay alive. All right, so these are the three things that we're going to talk about today. We're talking about diffusion, facilitated diffusion, and then osmosis. Okay, so by the end of this slideshow, you should be able to understand and define all three of those words. All right, and so passive transport is the first thing we're going to start with. Passive transport is the process that does not require energy, hence passive. Passive means you're kind of laying back, you're not doing anything. It doesn't require any energy when you're being passive, okay, to move molecules from high to low concentration. So I want you to think of molecules in a lot of them in a small area, and then dispersing out to areas that don't have as many molecules. Okay, so we'll talk about it as we go, but passive, remember we don't use energy. So diffusion is the movement of really small particles across a selectively permeable membrane. Okay, so the membrane is going to choose what it's going to let in and out. Okay, the cell membrane, as soon as equilibrium is reached, then we're good to go. Okay, so these particles move from an area of high concentration to low concentration. So high means just a lot more particles, whereas low concentrations means a little bit of particles. And once we reach equilibrium, that means we're going to have the same amount of particles on each side, and they're not going to flow either way. Okay, so here in this example, we have a couple different particles. We got 1, 2, 3, 4, 5, 6, 7, 8 particles on the outside. And we have 3, 4 particles on the inside. So we need to balance this out. And so we're going to go, this is our area of high concentration, and we need to go to low concentration. Okay, so we're going to flow this way. Okay, and we're probably going to have about 2 particles at least. So then that way we have 6 on each side. Okay, then it reaches equilibrium. So we're going to have particles flow into the cell. Okay, this is our inside of our cell. I'm just going to move myself over here. Okay, it's going to flow into the cell from the outside because we have a high concentration in here. Now this could flip. Okay, we could have had a high concentration on the inside of the cell and have it flow to the outside. So you got to keep that together. All right, let's just erase everything. Going on to our next slide. Here we go. Come on, one more. There we go. So diffusion. You can see we start in an area of high concentration when it's all in the corner here. High concentration, and we go to low concentration until we're all spread out, until we're completely spread out. This is a super important concept. Okay, going from high concentration to low concentration. But we see this all the time, you guys. We see this all the time. So if you ever had food coloring dye, can you put it in water? It starts out like this. It starts out with all the particles bunched up in the corner, and they slowly spread out to every single part of our slide. Okay, so diffusion from high to low. Now let's talk about osmosis. Okay, so osmosis is the diffusion of water through a selectively permeable membrane. So it's literally the same thing as diffusion, okay? But now it's specific to what? It's specific to water. Okay, now we're talking specifically about water molecules because water is so important that it gets its own type of diffusion called osmosis. Okay, water diffusion, or water diffuses across the membrane from an area, again, of high to low. Okay, we go from high concentration to low concentration. Okay, one way that I like to think about it, you guys, is I like to think about it as a hill. Okay, the high part of the hill, okay, is where we start, and we want to go to the low. All right, because of gravity, things that start on top of the hill are going to roll down, and they're going to go to low. All right. Now, if you're riding your bike, okay, you're riding your bike down a hill, okay, that's my beautiful bike that I drew. Do you need to pedal if you're going down a hill? You don't need to pedal, all right? That's why it's passive. Passive transport. Okay, passive transport. You don't need to pedal because the bike is just going to naturally go down the hill because you're at an area of high, you're at the top of the hill. Whereas if you wanted to pedal from low to high, then you would have to pedal your bike. Okay, you'd probably have to walk your bike up there. You'd have to put in some energy. Okay, we're going to talk more about that later. But let's go to this right now. So as you can see, this is really important. Looking at our little example here, but we have an area of, I wonder if I can pause it. But the water is flowing from high concentration to low concentration because there is the sugar molecules, as you can see, sugar molecules in here. Okay, that means there are less space for water molecules, and so that's why water is going to flow from high concentration because there's more water here than there is over here. Okay, so the water is going to flow from high concentration to low concentration. You just got to basically count the little particles. Okay, all right. So we're talking about our lipid bilayer. We have our cell membrane here, and then over time, let's count the particles. We have a bunch of particles here, and we have 0 here. Okay, so when we're thinking about high to low, you can write that. You can write high here, and we got low here. So it's going to go from high to low if we're talking about diffusion or osmosis. We have a high concentration up top and a low concentration down. So we're going to go from high to low. So some pieces, if it's water molecules, remember it's osmosis. If it's diffusion, so it's not water molecules, then it's something else. Okay, and so we have a few that cross over, and then eventually we have the same on each side. Okay, we have the same on each side. You could count them. I'm not going to, but we now have reached equilibrium. That's what everything wants to get to, is equilibrium. There's the same amount on each side. Okay, so again, we're not gonna spend a lot of time talking about hypotonic and hypertonic, but basically what you need to understand is wherever there is more particles. So if you put salt in water, if you put sugar in water, if you put anything in water, because you put the sugar and the salt in there, all right, so the sugar and the salt could be any of these little purple dots. Okay, because there's more purple dots on this side, there's less room for the water to be in there. Okay, so in this example, our high concentration of water is on the left. okay, because there's less things to take up the concentration of water. And so we're going to flow from high concentration over here to low concentration, okay, the low concentration of water. And so that's why we end up with more, it looks like more water on the right, because this side actually had the low concentration of water. So we're going to go from high to low. All right. So now we're going on to facilitated diffusion. Facilitated diffusion, think of facilitated meaning help is given. Okay. So facilitated, we're talking about it needs help. It needs help. Okay. And so that help is actually going to be these protein channels, okay, which are going to open up and allow things to go from low concentration to high concentration. Okay. And so the movement of larger molecules like glucose through the cell membrane, larger molecules, they have to be helped. All right. Protein cell membrane form channels for large molecules. So the facilitated diffusion, they're so big they need a protein channel, as you can see right here, in order to get inside of the cell. So they just need some help. Okay, let's keep on going. Let me erase this. All right. So you can see we have all these different protein channels. Right here is our protein channel that's going to let things into the cell. And then we have carrier proteins as well. We're going to talk about those in a second. All right, so protein channels allow, it just has extra space for the big molecules to fit in. All right, we're not really going to talk about hypertonic and hypotonic too much, but hyper contains high concentration, because think of when you're super hyper in energy, and then hypo is low concentration. Hypo, like you're taking a nap, you're bloated, you feel really big, hypo. Okay, and then isotonic means they're the same concentration. Okay, so hypotonic means the solution doesn't have very many concentration. Iso is the same, and then hyper means there's a ton of whatever you're trying to get inside the cell on the outside. All right, how this works with blood cells, just really quickly, hypertonic means that we are going to actually lose the water in our cells. Okay. Whereas iso, we're perfect. There's a good balance of both. And then hypo means our cells are going to blow up to a very high amount, and then they might burst. So that can be an issue. All right. Last thing we're going to talk about is active transport. Active now, if we were to draw our hill, okay, remember if we have that hill, usually we like to go from high to low. Okay, now we're actually trying to go from low to high. Okay, if we go from low to high, we call that active. Because if you ride your bike up a hill, you have to put in a lot of energy to get up that hill. Okay, you can't just let it ride and it'll bring you up the hill. You have to put in energy. And so because you put in energy, we call it active transport. Okay, energy is required, and you're pumping against what it wants to do. It wants to go from high to low, right? But you're pumping against it. Okay, so going from low to high costs a lot of energy, but it is possible to do. All right, let's just erase stuff. Let's keep on going. Okay, so passive transport. All that's happening is facilitated means it's big molecules going through the cell membrane. Diffusion is small molecules. Osmosis is just water. Okay, whereas active transport requires ATP energy. Okay, go ahead and watch this YouTube video if you want an example and kind of see it all in action. All right, my best analogy for you is you got these hills. All right, if you're riding your bike and you're going down the hill, then it's not going to require any energy. You just let it go and you're going to ride down the hill because you're going from high concentration, okay, to low concentration. All right, again, I would draw this out. All right, you're going from high to low. Okay, so passive transport. Okay, whereas when we flip it and we go from low to high, okay, Now it's going to be active. And energy is needed. You're going uphill. Okay, so active transport. All right, you guys are doing awesome. Let's see. I think that's all we got. Yeah, don't worry about endocytosis. All that's doing is really, really, really big particles. The cell membrane is going to wrap around it, and it's going to create a vesicle to move around the cell. Okay, this is how a lot of different cells are going to eat things, or how the lysosomes are going to grab things as well, so they can digest stuff. All right, so that's all we got for today. Make sure to figure out what active and passive transport is. Remember, the big thing we need to walk away with is going to be osmosis, passive transport, active transport, diffusion, and facilitated diffusion. If you know what all 5 of those different things are and what they mean, you can draw that hill and write them down. You're going to be just fine and ready for the test. Okay? All right. I'll see y'all later. Peace.